This is Simon and I'm Kaita, and our project is Resilient Rotifers. We're Year 12s from Tawa College.
So the aim of our investigation is to find an accurate and efficient method of water testing using the quantity, appearance and health of rotifers found inside it. So to do this, we're going to look at various water qualities such as salinity, water levels, nutrients levels, O2 levels, pH levels, etc. And we're going to see how these qualities of water affect the health, size, colour, shape, behaviour of the rotifers found inside of it.
So what is a rotifer? Rotifers are microscopic aquatic invertebrates which come in a variety of species. Rotifers come from the Rotifera phylum and are found in many freshwater sources. Rotifers are occasionally found in places such as seawater or rivers also. Rotifers feed off detritus, which is organic matter and all the tiny bits and bobs which are left over by other microorganisms, and by doing this they keep their environment very clean and healthy and prevent the buildup of mold, and because of this they are pretty vital in macro ecosystems worldwide. When it comes to a rotifer, there are three main parts inside a rotifer's body. The first part consists of the head, the middle part consists of the trunk, and the last part is the tail. Rotifers are very resilient creatures with specific adaptations which allow them to survive in a variety of extreme conditions. The main two adaptations rotifers have are anhydrobiosis and parthenogenesis. Anhydrobiosis is a process in the rotifer can remove 80% of its water content in order to go in a state of hibernation. This lets it basically sleep through extreme conditions. Arthenogenesis is a form of asexual reproduction that rotifers have adapted to perform. This means that they don't need a male to come along and fertilize the female zygote. This is very useful because rotifers can produce offspring at an immense rate and populations just increase and increase. As a downside to this, there's very little genetic variation as there's no chromosomal shuffling. But rotifers have also adapted to able to acquire DNA from the food that they digest, which is basically the counter to this, and this is what makes rotifers very resilient.
So now that we had a bit of background knowledge about how amazing rotifers were and how all the ways they could survive in all these conditions, we were ready to start planning our investigation. What we decided to do was to take 10 samples from a range of different water sources and then count the amount of rotifers in them and take notes about what they look like. We then repeated this 5 times. So Kaita will now go over the exact details of our investigation.
In our investigation we did many things to ensure the accuracy of our data and the validity of it. Some of these things were keeping certain variables controlled. This was like counting the rotifers in the same way, preparing the slides the same way, making sure the equipment was clean, and all these things. If we kept them controlled, it was going to be accurate. So we had many of these controlled variables. Our independent variable, as stated by Simon, was the different area that we took a sample from. The dependent variable was the amount of rotifers counted in it, and we had certain accuracy techniques like averaging the results to remove outliers and watching out for errors. As well as counting the number of rotifers, we actually looked at the conditions of rotifers and the conditions of the sample. This meant that we didn't have just this quantity data, and we could actually make connections between the environmental factors and the characteristics of the rotifers.
So the 8 different places we took water from was some residential moss, some water from the river where it connected to the estuary, some water from mud, some water from some rock pools, some seawater, some water from Kaita's drain, some moss near the sea, and also some still pond water. We think this is a good large range of different conditions and will give us a good idea of how different conditions affect rotifers.
And to pop in, one of the things we made sure was kept the same, that we used the same population and the same sample was used after 3 hours, so it's all consistent. Regarding the conditions data, we took the temperature with a thermometer, we measured the average pH with homemade pH indicator. Salinity levels was the only one based off research, but other than that we did O2 levels. We also did nutrients and water levels through processes of straining and measuring how much water actually came out of like a certain volume of the sample.
Here on the screen is all the data we collected, and in fact Kaita searched more than nearly 130,000 square millimeters of microscope slides searching for rotifers. So huge kudos there. Here's a table showing the various conditions that the rotifers were found in their various water conditions. Using these results, we've created a couple of graphs to help us decipher the patterns in the data and be able to construct this water testing method that we were looking for.
Through the quantity data and the different conditions we found certain patterns. Looking at the graphs, we also found certain patterns, and from this we constructed this this line on this graph up here, which shows the optimum conditions for rotifers.
So by analysing all this data, it's clear that the basic environmental factors we tested are indeed very influential when it comes to the health and quantity of rotifers. Since the quantity of rotifers is generally an accurate reflection of how healthy a population is, we can easily see which water seems to support more healthy ecosystems. And our hypothesis does seem to be accurate. We have found that the optimal conditions for rotifers consist of warm temperatures, low salinity levels, high water levels, high nutrients, medium oxygen and a neutral pH. These conditions increase the efficiency and performance of rotifers' adaptations and functions, allowing rotifers to reproduce quickly and stay healthy.
Because rotifers are very resilient microorganisms, they're commonly found in a variety of places as shown in our investigation. Taking this into account, along with their fast reproductive rates and physical variety, this makes rotifers extremely reactive, but also reflective of their surrounding environment. Because of this, we think that rotifers will be perfect as a bioindicator for the health or the well-being of a macro ecosystem.
Through trial and error and much data scrutinizing, we are able to construct a basic flowchart that will tell the person using it what the pH, O2, salinity and nutrient levels of the water they are testing is likely to be. Obviously there's still loopholes in this and it is not 100% accurate. However, we believe that it is an important first step in what could be a very useful and interesting way to determine water quality. In order to use this flowchart, users would have to have some form of microscope. Although luckily rotifers are not actually too small in the scheme of things, and their microscope could be relatively rudimentary. Here's the flowchart we've constructed. So you have these orange boxes as like a question or something that you will answer, which will take you to the next oblong shaped box. These also have assisted with extra information that can help you to follow it and see if you're on the right track. And then finally you'll end up at a little pink box, and this tells you which water source your rotifers most match up to in terms of our collection of water sources. Obviously there's only a small limit, like a small range we have, but it will give you a rough idea of what the various water qualities are likely to be.
Reflecting on our investigation, we think that next time we should look at other microbes as well as rotifers. Rotifers are such resilient creatures as emphasized, but this may mean that in certain situations they may be present when other microbes may have not survived in extreme conditions. Because of this, there might not be an accurate representation of the macro ecosystem that you'd be studying, and it'd be useful to have other microbes to see the overall effect of environmental conditions on these macro ecosystems. Another factor we could improve on is to look at different rotifer species. There are thousands of rotifer species, and it would be interesting to see if other species react differently to these environmental conditions, or if other species are only found in New Zealand, and etc.
Yeah, and as with most scientific studies, this experiment has left us with more questions than we started with in some ways. It'd be interesting and very helpful for our water testing method to pinpoint some particular water qualities and see how these affect rotifers individually. Because it's still not 100% clear, for example, what causes the colour changes and various other differences. So it'd be really interesting to maybe just change the salinity in a thing of water and then see how the rotifers last over like a number of weeks or something. So this could be an interesting follow-up experiment to see what happens. So we'd like to thank our science technician who helped us identify the Rotifera species, and Miss Boyd who helped us to organise it all and signed us up.